Hello, this is Miss Dagenford, and this is part two of our weekend homework, and it is a continuation of our cell cycle discussion. Now, the cell cycle, we saw the G1, S, G2, and then mitosis. So collectively, we call G1, S, and G2 interphase. And we include all of those three to kind of represent normal cell activity. Although, as we saw from the previous video, S and G2 are really preparing for cell division. So what you're going to start seeing is a lot of organization for the cell, for the nucleus especially. And mitosis is all about organizing those chromosomes. And if you think about it, if I have 23 pairs of chromosomes and they're duplicated, I want to make sure that each of those daughter cells will have all of the chromosomes it needs. So if we start out with 4, each daughter cell needs to have 4. Now this is easily accomplished because now we've duplicated the chromosomes. So let's take a look at the 4 phases. Now we have prophase, metaphase, anaphase, and telophase. Now, some students like to do flashcards. I don't think you need to. I just think you need to have things very well organized in your notes. One thing you can do, if you'd like another way to study, you can take your notebook page where you're taking notes and you can fold it and you can kind of make yourself some easy flashcards without having to cut them up. That's one thing you can do. So let's go ahead and get started. Prophase is the beginning of mitosis. It does have a lot of things going on. And I kept things very similar between each phase. We have chromosomes, nuclear envelope, spindle fibers, and then a part that we haven't talked about yet, and those are the centrioles. So the chromosomes, the most important thing that's happening to the chromosomes at this point, they are coiling around big proteins. Now, they already do coil around proteins to some degree, but we need to make a super organized chromosome. So imagine taking this really long linear chromosome and we start spooling it up and spooling it up and winding it up and organizing it, so much so that we take something this long and we package it into a really thick, shortened version. Now, one thing I did forget is that we need to have a duplicated chromosome. So let's add another double helix to this. So keeping my colors distinct, what we would see is one side is going to represent one sister chromatid, and it's going to be really well coiled. And then we're going to have another side that is going to be really, really tightly coiled for the other sister chromatid. Now, cells can have different looks. These chromosomes can have different looks. You could have really short chromosomes, long ones. The centromere can be way up here. The centromere can be way down there. So there are some different varieties of them. But the point is the chromosomes now have to coil. And some books will use the phrase condense. So if I slip and say condense, that's the same thing. So each chromosome condenses into this really super coiled system. And that's when we see the chromosomes most visible under the microscope. Before, the chromosomes are there, but they're just really hard to see. They're just all of these little thin thread-like structures. So in interphase, the chromosomes are all just tangled up. They're still there, but you can't really tell. In prophase, the chromosomes in the nucleus are so coiled, we can start to see them sometimes better than other times, but we start to see them separate out from each other. Now, remember, mitosis is all about organizing. So the nuclear envelope, think about if you're organizing your desk, you might clear off everything, and you might open up your room and put stuff in the hallway, and so you can think about it that way. The nuclear envelope actually breaks down. Now, it doesn't disappear in the sense that we never see it again, but we don't see it as an organized structure. It just separates apart. And so we don't see it under the microscope anymore. And that's to kind of use this very organized system. The spindle fibers. Now, the spindle fibers, that's an old word. They are actually microtubules. Sometimes you will hear them called the spindle apparatus. And a spindle was an old structure used in a loom, and it kind of had a structure that looks like this. That's not a very good picture. But in a cell, these microtubules, they form little strings, very visible strings under the microscope. And when you see them under the microscope, notice that it sort of looks like this structure. So we call that's how that's how it got its name. But really, you need to understand that these are microtubules. They're going to form this cage around the chromosomes, and it's going to allow you to distribute the chromosomes evenly. Now, what's doing this is an organelle called the centrioles, and I like an older term that we used to call it, called microtubule organizing center. Too big of a mouthful, didn't catch on. They themselves are made of microtubules, and they have this weird little circular shape, and it looks like it has grooves in it. There are two centrioles, and they tend to be at perpendicular to each other, and then they're made of microtubules, and they help organize the spindle fibers. Usually they are right outside of the nuclear envelope, but when the mitosis starts, the centrioles divide, and then the pairs migrate to opposite ends. So if we were to real quickly draw this, we would show the nuclear envelope breaking down, and then the centrioles, which I'm just going to draw as these little rods here, they're migrating around until they're opposite each other. In the meantime, the chromosomes are going to start coiling up. Now, I'm just going to draw 4 to make our lives easy, and that would be an artistic version of prophase. Now, the other ones are much easier because a lot of the things stay the same. So for instance, the nuclear envelope in metaphase, the nuclear envelope is gone. We don't see it. The centrioles are at opposite ends, and we call those the poles, like the North Pole and South Pole. The interesting thing in metaphase are the chromosomes and the spindle fibers. The chromosomes attach to the microtubules. Well, they are attached to the microtubules. And the spindle fibers move the chromosomes to the equator of the cell. So let's draw that out. Here's our cell. The nuclear envelope is now gone. I'm going to use red for the chromosomes just to make it a little easier to see. And so the 4 chromosomes line up in the middle. And the microtubules help this happen. They tug and tug and tug until the chromosomes are all lined up. Now, you may well ask, how in the world is this happening? That's a good question. There are signaling pathways that help this happen. And some of it's still a mystery. We're still, working things out. So here are the centrioles at opposite ends. And then we have our microtubules, the spindle fibers that are creating that cage that spindle apparatus. And I'm just drawing dotted lines to kind of make this a little bit easier to see. If I slather my drawing with lines, it starts getting really hard to see. And so these microtubules are attached to the centromere of the chromosomes. Now, again, remember I said at this point, we're really just looking at the chromosomes. The nuclear envelope's still gone. The centrioles are at the pole still. So that's going to be the same. The nuclear envelope is gone. The centrioles are at the poles. So what's happening to the chromosomes? Well, the chromosomes are pulled apart at the centromere, and the spindle fibers are help pull them apart. And then there are these little motor proteins that start walking the chromosomes towards the pole. So the spindle fibers and the chromosomes walk the chromosomes to the poles. So let's draw this out here. I'll go over here. So let me go back to my black pen. And our centrioles are still at the poles. And we still have our spindle apparatus, our microtubules. Now, this is big key. The chromosomes have been pulled apart. And as they pull apart, they're not solid structures. So think about pulling a string in the middle. If you pull a string at the middle, both ends kind of fall back. And that's kind of what I'm indicating here. We'll watch a video that you'll be able to go, oh, I see. So the chromosome's been pulled apart. Now at this point, we are calling each one of these a chromosome. And so that's a chromosome and that's a chromosome. And notice at this point, we now have 8 chromosomes, 4 of each kind. But remember, our purpose is to create a duplicate set of chromosomes. And now we've done so. We've pulled it apart. Now, the only thing that we need to do now is complete. So the chromosomes reach the centrioles in telophase, and they get pulled in really tight. The nuclear envelope forms around the chromosomes, and so telophase is the exact inverse of prophase, everything that happened before. So the chromosomes reach the centrioles, and let's also not forget that now they have to uncoil back into chromatin. The spindle fibers break down because we don't need them anymore. And the centrioles are there until the cell divides next. Now, the other thing we need to talk about is what happens to the cell. We call that cytokinesis. The membrane pinches in, and then eventually it'll pinch in completely, and lo and behold, we've got a new set of daughter cells. Now, an onion, in plant cells, because they have a cell wall, they need to grow a new cell wall between the two. So let's go ahead and draw it out. Cytokinesis, here's our cell membrane kind of pinching in here. The nuclear envelope reforms around each of our set of chromosomes. The centrioles are outside the nucleus. And lo and behold, we have our 1, because it's uncoiled now, 2, 3, 4, and then the other set, 1, 2, 3, 4. And happy, now we've got our 2 cells, each with 4 chromosomes. I hope that helps. We're going to practice this on Monday.